So again, the enzymes are special proteins, and their only function is to speed up chemical reactions. And biologically, that's important because you want to make sure, at least organisms and cells want to make sure they have enough product produced by the chemical reactions that the enzymes are accelerating, made to go faster and faster and faster. Okay? Without enzymes, most chemical reactions occur too slowly. And in this example, you wouldn't supply enough blood sugar, glucose, to your body, to your cells. So the way that enzymes speed up the chemical reactions is by doing something called reducing the required activation energy. So the activation energy is the energy that's required to start a chemical reaction. So to break this chemical bond, there has to be an input of some energy before the chemical reaction can occur, meaning the breaking of the bond. It's like if you're going to strike a match, you have to supply some energy to, push the match against the little strip so that the fire gets started. So in this case, the activation energy is the energy required for a certain particular chemical reaction to occur. In this example, to get the starting reactant material, sometimes called a substrate, into the end product materials. Okay? Now this is without an enzyme, and the activation energy is whatever it is. Okay? If we add an enzyme, the exact chemical reaction is sped up, accelerated, because the enzyme reduces the activation energy that's required for the same chemical reaction to occur. Since the enzyme reduces the required activation energy, the reaction can occur faster, right? And then the enzyme can latch onto another reactant molecule and have the process occur faster, faster, faster. Okay? So enzymes work by lowering the required activation energy for a particular reaction to occur. It's like, let's say this was a boulder, and you needed to push it over a hill. It's easier and faster to push the boulder over a small little hill than it is a big hill. Okay? All right. So the enzyme, all it does is latches onto the reactant or substrate molecule. In doing so, reduces the activation energy so that the chemical reaction can occur usually much, much faster. Okay? So they reduce the activation energy required for a particular chemical reaction. So enzymes have what's called substrate specificity, or they're specific for a particular substrate. Again, we can call a substrate a reactant molecule. Right, so only certain enzymes will work on certain specific substrate. Notice this example, lactose, milk sugar, is acted upon the enzyme called lactase. So this enzyme will only latch onto, bind with lactose. It's that specific, it won't bind to any other sugars, and help speed up the chemical reaction, in this case the breakdown of lactose into glucose and galactose. Okay? And the reason it has substrate specificity, or enzymes have substrate specificity, is because of their shapes. They have specific active sites that fit the specific substrate. Again, kind of that analogy to a lock and key or a hand in a glove. Right? And notice the name of this particular enzyme, it's called lactase. Most enzymes have a ASE ending, and often the name gives you a sense of what the substrate or reactant molecule is. Lact for lactose, ACE for the enzyme that latches on to and speeds up the chemical breakdown of lactose. Okay? So after the product are released, the enzyme can bind onto another substrate molecule and just do it again and again and again. And again, many enzymes are named for their substrates and have that ASE ending, like lactase enzyme that acts upon lactose, its specific substrate. So if you can break down and utilize lactose in milk, it's because you produce in your body the enzyme lactase. Some people who are lactose intolerant don't produce the enzyme lactase to break down lactose, and then lactose causes them some digestive issue problems. Luckily, it looks like Kermit has lactase produced in his body. It's Kermit the Frog, by the way. Okay, so how enzymes work, a little video here.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy.
And he's using some fancy terminology, exergonic reactions. Don't worry about that. Just realize that the starting substrate or reactant molecules do have higher energy than the ending product molecules, but in order for this to occur, this chemical reaction, there has to be that activation energy that has to be inputted to get the chemical reaction to move along, to proceed. And an enzyme works because it lowers this activation energy, allowing the reaction to occur faster and easier, if you will, but faster.
How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
All right? Okay. So let's look a little bit on the function of enzymes on our biological membranes. So cells can regulate the movement of material in and out of cells by regulating how they move across the membrane. And when we talked about biological membranes, we did talk about how some things can simply diffuse across, but some things can't, either because they're charged, they're ions, or because they're too large to easily diffuse across. And here's where membrane transport proteins come into play. They're channels, and some of them will work to move things in and out of cells. Some proteins react as signaling proteins, and they respond to some chemical in the environment and send off a signal inside the cell. And then notice here, some proteins are enzymes that are on the membrane, and they act in speeding up chemical reactions. So, and notice the different shapes of these proteins indicating their different functions. Okay, but notice that some membrane proteins are enzymes or have enzyme activity. A little bit on membrane selectivity here.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Okay, and that's where membrane proteins as channels come into play, because those membrane proteins can be channels, passageways for things like glucose, blood sugar, and certain ions to move into or out of cells. Another one. I think.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells?
So this is the phospholipid bilayer zooming into one cell in your small intestine, your lining of your gut that the food molecules have to get across. So that's outside the cell where all the food stuff is that's been broken down and digested. There's the phospholipid bilayer. These are membrane proteins. And this is inside the cell.
Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly.
And I can't remember if the narrator mentions it as well, but glucose, blood sugar, also enters into a cell by facilitated diffusion.
The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
So a little bit of everything there, but focusing on membrane proteins and how they can function as channels to do facilitated diffusion, which is a passive process, or active transport, which requires energy, and the example there was the sodium-potassium pump. And of course, some things can simply diffuse across the plasma membrane, like carbon dioxide and oxygen. And lipids, fats. Okay, we'll continue on our next slide, next lecture.